Hi, I'm Dr. Ben Kroeger, and I work in Kieran Harvey's lab here at the Peter Mac. As part of our latest research, we've captured something new on film for the very first time. We've been studying the Hippo signaling pathway, which is an ancient cell signaling pathway that controls whether cells grow, divide to form two new cells, or die. We've captured microscopy footage showing the behavior of DNA binding proteins of the Hippo pathway as they explore human cancer cells and bind to DNA. In red are the molecules, and in gray is the DNA, which is mostly found in the nucleus of the cell. In this video, you can see on the left is TEAD, which is a transcription factor, moving around through the nucleus. Some molecules finding and binding to specific sites for numerous seconds. Whilst on the right is YAP, and YAP is found both in and outside the nucleus. And we studied the molecules within the nucleus as they go in and bind to DNA. TEAD is a transcription factor, and transcription factors are a special type of protein that bind to your DNA and allow and control a process called transcription, which is how the DNA code is read. So here we're looking more closely at TEAD, which is the transcription factor. On the left, you can see TEAD is found mostly in the nucleus, where it's moving around and binding to DNA. In the center, you can see a mutated form of TEAD that can't bind to DNA, and you'll notice all the red molecules are moving around much more drastically inside the cell. Whilst on the right, there's a mutated form of TEAD, which is found in a rare genetic disease that causes retinal degeneration. In this movie, we've imaged single molecules of TEAD as they move through the nucleus of a human cancer cell. And we've particularly focused on areas of the nucleus which have lots of YAP protein, which is what TEAD, the transcription factor, can bind to. And we were particularly interested in the behavior of TEAD in these small regions of the nucleus. So this is a map that I've created using the movies that you've just seen. We followed the movements of single molecules through time and plotted their position. And what you could see is different sorts of movement through the nucleus. Some molecules, shown here as squiggly lines, moved throughout the nucleus over the time, whereas others were still. See them more like a dot in here. And these are the molecules that we think are binding to DNA. Ultimately, we hope that some of the fundamental discoveries that we've made here will better inform our understanding of new treatments for several different types of cancer, including mesothelioma.